Brain cancers are the second most common kind of cancer in children, the first one being leukemia. And because it's a tumour, it's a solid mass tumour, it means that we can model that in our model systems, and it's also got a good survivability in human patients. So some forms of medulloblastoma, 80 to 90% of people survive into adulthood, and we're really interested in what happens to the survivors and the impact that the cancer treatment itself has on the rest of their lives. So why do we look in just children? It really boils down to the fact that our nervous system is still maturing during childhood. So, from birth all the way through adolescence, really, our brain and our peripheral nervous system are changing the way we process these things. And because it's growing and maturing, if you interfere in that process with, say, a cancer chemotherapy drug, you're going to change the way that it works when you're an adult. And that's why we're really focused on that. This piece of research is really designed to look at what cancer tumour cells produce during chemotherapy, and the factors, the chemicals that they release, and how those chemicals, those factors, interact with the bits of our nervous system that detect and process pain. What we think is that the chemotherapy drug, when it's doing its job of killing cancer cells, makes the cancer cells release things which also alter the way in which our nerve cells grow and mature. And that leads to a long-term change in the way that these people experience pain and their susceptibility to pain. We're isolating EVs from medulloblastoma cells, and we're analysing what they look like, how big they are, how small they are, and what they contain. And we're going to sequence, we're going to identify the chemicals that are within EVs and see if different chemotherapeutic drugs change what those EVs contain. We're really interested in the cargo in the EVs, what's inside them. And we've particularly got an interest in RNAs, so very small molecules that are part of the genome. We think that these RNAs that come from cancer cells might reprogram peripheral nerve cells. If we identify an RNA that does, or a family of RNAs that change the way nerve cells work or mature, that we'd be able to interfere in that process with other RNA molecules working as drugs and stop that ever happening. What's the impact of the research? From a scientific point of view, it's trying to identify new ways of thinking about childhood cancer. And up until this point, most laboratory studies have focused on drugs interacting directly with the nerve cells, but the tumour's been forgotten entirely in this process. So actually a change in the way we think about childhood cancer and its treatment. The foundation funding is amazing. This research would never have happened without the Medical Research Foundation funding. It enabled us to come together as a group of scientists, of applicants on the grant, who'd never really worked together as a whole group before and focus on something that's never been tried before. It's something that we've all been desperate to do, and it would never have happened without the Medical Research Foundation funding.